variation. Continuous variation is described by some kind of continuous metric, which could be like inches or speed or enzyme capacity or all these kinds of things that can be measured on some kind of linear scale. Most morphological traits are like this. Here's a histogram of student height in some class, and you can see, it's this roughly normal type distribution where there's a few people that are really short, a whole bunch of people in the middle, and a few people that are really tall, and every possibility in between has some possibility of representation, so that the distribution of variation is effectively continuous. You have lots and lots of traits like this, and you get those because these traits are controlled by lots and lots of different genes, having each of which has a relatively small effect. So what you saw was a histogram of the phenotypes, right? As you know, the phenotype is due to the interactions of the genes with the environment during development, and that environment that genes are interacting with also includes other genes, right? It can be an internal as well as an external kind of environment. So the phenotypes that are expressed in organisms are, you shouldn't be thinking of them as entirely genetic or entirely environmental. Essentially all traits have some kind of combination of the variation in that trait being due to genetic variation and or the variation in that trait being due to, what did I say first? One's environmental, one's genetic. It's both is what I'm trying to say. So, and students often get this kind of confused, this idea of heritability versus inherited. There's all kinds of things that are inherited, meaning having some kind of genetic influence, that is separate conceptually from heritability. What we're talking about here is differences within populations being due to differences in genes, not just whether there is some genetic influence on a trait. So if you look at what are the kind of possible causes of variation, phenotypic variation, in some trait, in some population, this is usually broken down mathematically in this very sort of simplistic way where you say, oh, phenotypic variation VP has to be due, there's really only two sources of that, right? Genetic and environmental. And then you can break down the genetic sources of variation farther into the different kinds of genetic effects that you would have within an interacting genome that's developing. So the genetic effects can be due to what are called additive effects, and those are the ones that we'll talk about and focus on sort of for the rest of this lecture. But there are also epistatic interactions, there are also dominance genetic effects, there are maternal effects, there's epistatic bidominance interactions. There's a variety of other things, but what I want you to understand for now is what do we mean by additive genetic effects, and why are those the ones that kind of are more important for understanding quantitative trait evolution. So, additive effects mean that they're additive. So that lots of different genes have different separate effects that combine in a kind of additive way. They accumulate additively. What's an alternative to additive effects? Non-additive, multiplicative, right? You could have one where the effect of one trait would depend, the effect of one gene would depend on the alleles at a different gene, right? That would be a non-additive effect. That's epistasis, right? So additive effects suggest that they're acting separately from each other and accumulating. So I've made this little contrived example where you have 5 loci affecting some trait. Each has 2 alleles, so you have 10 alleles total. And what they do is they affect sort of the relative growth of some kind of trait, where each of these, the allele 1 has a negative effect. Like if you're growing a leg, allele 1 will make your leg a little bit shorter. And same thing with the B locus, and C and D and E, and these have kind of separate additive effects to make your legs a little bit shorter, right? And the alternative allele has some kind of effect to make your leg a little bit bigger. They're not necessarily leg genes in the sense of being bones and muscles and whatever, but they're developmental regulatory things that affect how long genes are turned on and all kinds of other things that will affect how big a leg you end up with, right? So if you start with each of these having a frequency of 0.5, so at the A locus, the two alleles are equally represented, and that's true for each of these, right? Then you can figure out what kind of phenotypes that you get, right? So that you understand this, how do you get the shortest phenotype, the -5 phenotype? There's only one way to do it, right? You have to get all 5 of the -1 alleles, right? So you have to get at A, B, C, D, and E loci the negative allele, right? If each of them has a probability of 50%, each one is at 0.5, then this is 0.5 times 0.5 times 0.5 times 0.5 times 0.5 is the probability of getting that phenotype, which if you do that really quickly in your head, that's 0.031, right? In other words, you would expect 50% of the population to have this shortest phenotype. Okay? Now, by contrast, how can you get the next smallest phenotype? You have 5 loci. Four negative, 1 positive. You're thinking exactly the right way. It doesn't matter which one of them is the positive one. No, right? You can have 1, 1, 1, 1, and 2, or 1, 1, 1, 2, and 1, 1, 1, 2, 1, 1, 1, 2, 1, 1, 1, 2, 1, 1, 1, 1, 1, 1, right? They're all equivalent in terms of producing that -3 phenotype. If all those alleles have a frequency of 0.5, then any one combination of them has a frequency of 3%, but this phenotype has 1, 2, 3, 4, 5 times 0.031, right? So what's the frequency of that phenotype in the population? 5 times 0.03. Right? 15%, right? The genetic architecture is exactly the same, and you have this, because you have one way of getting this one of the 5 loci, but it doesn't matter which one, this is a more common phenotype in the population. The -1 or the +1, both of those require 2 of the 5 loci, right? So there's a lot more combinations of ways to get those, right? It's any one phenotype has a frequency of 3%, but there's lots of ways, any one genotype has a frequency of 3%, but a phenotype, there's multiple ways of combining those. So these are more common, and of course, if you just take this out, there's only one way of getting the tallest phenotype, having the alleles 2, 2, 2, 2, and 2, right? So here you have a simple trait, 5 loci, 2 alleles each, and just by combining the probabilities of getting each of these alleles, you get a distribution of phenotypes that looks like that. Ooh. What does that look like? Looks like a normal distribution, right? It's this kind of effect which is why you tend to have normal distributions of continuously distributed quantitative traits. But this becomes important for a bunch of other reasons besides just that. It means that there can be a really big response to selection without any new genetic variation being involved. So this phenotype is really, really rare in the population. But let's suppose it's favored. Let's suppose there's selection, in this direction. Without losing any genetic variation, just changing the allele frequencies, you can have a big response to selection because of kind of the uncovering of hidden genetic variation that is wrapped up in these rare alleles. So if all 5 of those loci went from being 0.5 big and 0.5 little to being 0.6 big and 0.4 little, then you'd have a shift in the distribution like this. Doesn't look dramatically different, but now, just so everyone knows what's going on, this roots, this one, used to be at a frequency of 3%, 3.1%, because it was 0.5 to the 5th, right? Well, now it's 0.4 to the 5th, which I don't know what that is, but it's 0.4 times 0.4, right? It's less common than it was before. This one over here that used to also be at 3% is now at 0.6 to the 5th. It's become more common, not due to any new kinds of genetic variation, just due to the reshuffling of the existing genetic variation. And you can do this kind of thing for a pretty long way before you lose genetic variation. So if you just extend this out to over here, say, you have at all loci, the big allele has a frequency of 0.8 and the little allele has a frequency of 0.2. And the phenotypic distribution is a long way from what it was before, just by recombining genotypes and changing the probabilities of getting those different additive alleles. So this one has a frequency of 0.2 to the 5th, and you don't even see anybody there. There's still genetic variation that phenotype could be produced by that population. It's just extremely rare based on this kind of uncovering of existing genetic variation. All right, so you can take this even farther. As some alleles reach, some loci reach fixation of, say, the larger allele, you can get, huge amounts of phenotypic change from existing genetic variation, and it's pushing around this kind of normal distribution. So does everyone see that, how you just kind of multiplicatively combine the probabilities of these different alleles joining together to produce these phenotypes, and why that gives you this kind of distribution? Okay, you made me happy. All right, so that's going to affect how much the potential for an evolutionary response to selection. So remember, evolution by natural selection is change in populations over generations. Selection happens within a generation, and evolutionary change occurs across generations. It happens if individuals vary, some of that variation is genetic, there's differential reproduction that is non-random with respect to these genetically varying traits, right? So to get evolutionary change, you have to have genetic variation. To have selection, you don't have to have genetic variation, right? Which one of these is selection? Reproduction being non-random is selection. You can have that without getting evolutionary change. You have to have these other things as well. You have to have this kind of genetic variation. So that's where heritability formally comes into this, and that's the fraction or proportion of phenotypic variation that's due to additive genetic variation. So heritability is symbolized as h squared. It's VA, additive genetic variation, divided by phenotypic variation. Ranges from 0 to 1. It's a variance term, which is why it's squared. You don't have to square any of these numbers. That's just what it is. And you can estimate it from the resemblance between relatives. We talked about this 10 days ago, something like that, right? If you have the average of the offspring and regress that on the average of the parents, then the slope of that line equals the heritability, right? There's lots of other ways you can do it, but this is the easiest to visualize. So if you look at these graphs, well, you can see here the slope is not different from 0. So that's telling you the heritability is 0. There's still phenotypic variation there, but it's due to something other than additive genetic variation, probably mostly the environment, right? Here's a slope of 0.5, here's a slope of 1. It's not that important how much scatter there is around this. Students often confuse the tightness of the data to the line with the slope of the line. For heritability, we're interested in the slope of the line, not how tightly the data points cluster on it. That slope is telling you how much offspring resemble parents because of average additive genetic effects. And again, it ranges from 0 to 1. If you're interested in measuring selection to predict an evolutionary response, you have this selection differential F. And what that is the difference between the average traits of those that are reproducing in any one generation and the entire population in that generation. So again, selection acts on phenotypes within a generation. And F is the difference between those that are reproducing and the average of everybody. And it's in the units of the trait, right? So if there's selection on running speed, just stick this over here for a second. So you have number of individuals. Here's how fast they run. It'll be in like meters per second, because you all use meters now, right? Here's this is the distribution of the entire population, and it has an average of this, whatever that is, 2 meters per second. And here's those that get away from the cheetah or whatever and actually reproduce, and their average is, say, here at maybe 3 meters per second. What's F? It's the difference between them. So F equals 1 meter per second. It keeps those units, right? What if ones that were slower were favored? What if these ones were favored? Over here, they could only run 1. For whatever reason, slower is better. What would F be? It would be -1 meter per second, right? So it's in the units of the trait that you're looking at, which is great, except what if we want to compare strength of selection on running speed in U with like rate of ear growth in elephants, with digestive efficiency of mice? We're interested in comparing strength of selection a bunch, all different kinds of stuff. But we have this metric F that is in units of the trait. That's not going to be very comparable across studies in organisms and traits, right? So an alternative to that is this thing called the selection gradient, which is the slope of relative fitness on the trait value. So this cancels out the units, and you get a metric that's comparable across all different kinds of studies. It's still called F, but it's in these kinds of equations. So an alternative way to calculate selection gradient is the selection differential, S, divided by the trait variance, right? So selection differential is in meters per second or whatever, and the trait variance is in meters per second. So you get this unitless kind of thing. So here's an example with, selecting for longer tails in mice. Right? Here's the whole population. Here's the average of the whole population. Here's the average of those that breed. It's that, and that difference is F. Okay? Yeah.
When you said the negative value that was 1 minus 2 and you got minus 1, so what is the negative sign?
It means the selection is favoring those that are below the mean. Shorter tails or slower running speeds or whatever. Right? It really is the simple difference between those that are reproducing and everybody.
So if it's positive, then the selection is favoring those above the mean, and if it's negative, it's below the mean.
Right. We're going to talk about modes of selection in a minute. They're both directional selection, right? So F, I don't like green anymore. If F is positive, that's directional selection going this way, right? And if F is negative, that's directional selection going that way. It really is just numerically that difference. Okay. So, again, this kind of theme of evolution being predictable in some sense, we can calculate a predicted response to selection if you know a couple properties of a population. So a response to selection is evolutionary change across generations, right? And it's R, and how much you expect the population to change from the parental generation to the offspring generation depends on how strong selection is, seems intuitive, and on the heritability. And that also ought to be intuitive, because what is it that links the parent generation to the offspring generation in terms of their trait values is genetics, right? So the offspring are only going to differ from their parents to the degree that what was selected upon had some basis in genetics. So if the heritability was 1, then the response to selection is the same as the selection differential. If that variation in the parental generation is entirely due to genetic variation, and you select for certain variants of the parental variation, again, due entirely to genetic variation, you expect the kid generation to exactly mirror what was favored in the parents, right? Because it's linked perfectly. If the heritability is 0, then the response to selection is 0. So this is the obvious kind of case where can you have selection and not have evolution by natural selection? And the answer is yeah. Selection and the response to selection are separate things, right? The response is this change over time. Heritabilities are usually intermediate, and the response is kind of proportional to that. Oh, before we get to this, let's just kind of draw this out a little bit. So what you have this is the parents, right? They have some phenotypic variation. Here's the mean of the whole population, right? And I don't like green, you guys can't see it. Here's the ones that reproduce over here. Right? So let's say this is 5 and this is 2. What's F? Okay? So there's directional selection for less of whatever this thing is. That's selection on phenotypes in one generation, right? So then you have to get to what are the kids going to look like? And this is going across generations. Right? Well, we would expect the kids to have shifted to the left because that's what was favored in the parental generation, right? the extent that they do so depends on heritability. So let's suppose h squared equals 0.5. Suppose we know that for whatever reason. We did a study and we figured out empirically that's what it is, right? What's the distribution of phenotypes in the kid generation going to look like? What's the average going to be? It'll be 50% of what? Well, it's all about the trait that's favored, right? F was -3, so the R equals h squared f. R equals 0.5 times -3, which is -1.5. So now, what's the average of the kid generation going to be? You with me? Right? So you can predict quantitatively, you know, you're trying to like take chihuahuas and breed something that's actually worthwhile, and you measure their traits and you impose selection on it, and you want to be able to predict generation after generation how much change there is in the population. That's why this R equals h squared s is actually called the breeder's equation, because it was developed for artificial selection based on humans wanting to know, I'm trying to make my livestock better, how much of a response can I expect? Yeah. Okay, so we figured out that the R is -1.5, right? So then you have to say, what was the average of the parental population? It was 5. Yeah. Yep. So that R is the change in the population across generations. It's that evolutionary response, right? And then if you do this, it makes sense that all of this is in the units of the trait that you're talking about, because you end up with the average being, 3.5 whatever they were before. Right? So hopefully you can all kind of do little numbers like that in your head and do these kinds of problems. I just wanted to point out, though, when I gave you this example, I said, just suppose that heritability is 0.5, and you just happen to know that from some other study. But you should be able to look at this and figure out, you know what? I could figure out heritability by doing a selection experiment. Right? If you know what the parental population is, and you impose selection where you know what that is, and then you measure the response, what have you figured out? Right? If you have R equals h squared f, and you make f something that you want it to be, and you measure R, then you can calculate heritability. So in addition to parent-offspring regression and these other ways, this, doing an artificial selection experiment is one way to calculate what the heritability of some trait in some population is. Okay, so you should be able to do all those kinds of little back and forth calculations. Okay, next but related topic is these typical modes of selection. How does selection operate on these kind of quantitative continuous traits? Here's a bunch of graphs that talk about 3 main types. One is directional selection, stabilizing selection, and disruptive selection. And for each of them, they have these associated graphs that say probability of surviving, which really, who cares about survival? That's not what evolution is about. This ought to just be, fitness. So fitness as a function of the trait, if fitness as a function of the trait is going up like this, that means phenotypes on that extreme upper end are favored. That's why it's directional selection. But as we were talking about with when S is negative, you know, in red here now, this is also directional selection. It's just favoring the alternative extreme. So what that happens with selection is you get a shift in the average, and you usually get a reduction in variation. With stabilizing selection, you get no change in the mean, but you get greatly reduced variation around that mean. Okay? And you can kind of have, well, let me just finish disruptive. Disruptive selection means that individuals of the two extremes are favored, and it's worst to be in the middle somehow. So this favors ultimately some kind of distribution that's going to look, you know, bimodal. This is the kind of thing that we associate with speciation, right? Divergent selection, to be either this or that, but not in the middle, is what we conceive of there. Now, what hopefully you can kind of see is that these aren't completely open-ended necessarily. So if you have fitness as a function of the trait values, and here's the current average, right? If that's the fitness distribution, what would you call it? You go, oh, it's sort of stabilizing because look, these ones have low fitness and these ones have low fitness. But is it stabilizing for the current average? Here's the current population average, and who has the highest fitness is an average that's over here, right? So there is some degree of directional selection, but it's not open-ended, right? It's like, this is favoring you if you're bigger, but not too big, right? And that kind of, you know, presenting it as there are these three modes, directional, stabilizing, and disruptive, is definitely overly simplistic for most kinds of traits. In fact, most traits are going to have a fitness surface kind of like this, where there might be some directional selection, but there's a limit to it beyond which some other force is going to really push back, right? And that's what makes stabilizing selection the most common type of selection for most traits of most organisms. There's really kind of two different related reasons why this is so. Can anyone guess, like, why is stabilizing selection more common than directional or disruptive? Give me one of two major reasons. Nope. So stabilizing selection decreases variability, first of all. Second, things aren't here because of their effects on populations and species. Remember, we were talking about levels of selection. Things are the way they are because of their effects on the fitness of individuals, which should give you a hint of the question. The reason I'm getting at number one is, why are you here? I want to get a biology degree. Why are you here in the big sense? Because your ancestors reproduced, survived and reproduced, right? They did something that was good enough for their environment to allow them to survive and reproduce. That's why you're here. Their traits were good enough for their environment for them to reproduce. So why are most traits most of the time under stabilizing selection? Because they've been subjected to a long history of selection already. Most traits have already been optimized for major features of the environment. Now, if the environment changes radically, then that would change what the kind of best trait value is, right? But if the environment is relatively stable, then most traits are under stabilizing selection because they've already been optimized by selection to be good for that environment. That's major reason number one. Major reason number two is what I call trade-offs. Trade-offs that give you net stabilizing selection. So here's a picture of survival. So it's a picture of a plant. It's called a goldenrod. And this big lump on the stem is a gall made by an insect. An insect laid its egg in there. It's called the goldenrod gall fly. It lays an egg in there that induces this sort of cancerous growth in the plant, and then the insect larvae grows and develops inside there. And that insect larvae is attacked by a wasp. It's a parasitoid wasp that lays its egg on the insect larvae. And here's percent parasitism as a function of the size of the gall. This is gall size. And what you can see is if the gall is really small, you're very likely to be parasitized. And if the gall is big, you're not very likely to be parasitized. Right? So that's selection favoring larger gall size. Right? But this right panel, these goldenrod gall flies, the galls are also ripped open by woodpeckers that then eat the larvae that's developing inside. And here's the percent predation. Do woodpeckers rip open small galls? No, not really. Do they rip open big ones? Yeah, they rip open a lot. So you have these two different contexts of selection. One favors big galls and one favors small galls. There's a trade-off between the kind of trait that would be optimized just if there was wasps and the kind of trait that would be optimized just if there were woodpeckers. So what happens is there's net stabilizing selection between these two opposing forces. So there's selection from woodpeckers for smaller, and there's selection from wasps for bigger, and what you get is net stabilizing selection. And that's true in all kinds of different sorts of trade-offs, right? There might be, the kind of classic examples. The male peacock might have, a fitness as a function of tail length for these male peacocks, right? This might be due to mating success, right? Female peacocks prefer these huge, ridiculous tails. So in the context of mating, there might be directional selection for longer tails. But that might make you easier to be killed by a tiger. So predation might do this, right? So there might be opposition where sexual selection is directional this way and natural selection is directional that way, right? Different kinds of contexts, not just predation, right? All these things both give you this kind of net stabilizing selection. So you should think of natural selection working on traits as is not maximizing any part of fitness, right? What natural selection does is optimize each of the components. These components are under conflicting selection pressures. So that's major reason number two, that most things are under stabilizing selection. So that's modes of selection in kind of a part one and part two. So then the next big question, as you have this genetic architecture of traits that involve lots of different genes, as we're built up to this kind of more realistic model, the question becomes, does selection at one locus affect evolution at a different locus? And so far we've assumed they don't. We've assumed they're independent evolutionary kinds of trajectories. And the answer is it depends. Evolution at one locus or selection at one locus can affect selection at a different locus if they're linked, if there's some kind of linkage between them. And so linkage is, we already talked about chromosome inversions. What did we call the genes that are within a chromosome inversion? Come on, I know you were talking about it at a party last weekend. It's called the linkage group. What happens with a chromosome inversion? What does a chromosome inversion prevent? Prevents recombination. That's why these the alleles at these loci within a chromosome inversion, their fates are linked together because they're not broken apart by recombination, and so they don't have independent evolutionary trajectories, right? So that kind of chromosome inversion is an example of physical linkage, where the genes are close together on the same chromosome. They could be in an inversion, linked together much more permanently, or even if they're not in a chromosome inversion, if they're just right next to each other on the chromosome, what's the likelihood of recombination occurring between them? Not very good, compared to if they're really far apart on the same chromosome. It's much more likely that recombination will occur between them. All right? So that's kind of easier to visualize. You can also get linkage that is of genes that are on completely different chromosomes. It's not physical linkage at all. There's lots of different ways of getting linkage, but any statistical association between the alleles at loci that's non-random is linkage. And it can be from all kinds of different sorts of reasons. If I tell you that someone has blonde hair, do you know what their eye color is? No. But do you have a better than random guess? Because there's a non-random association between genes for eye color and genes for hair color, right? Okay, so to do this, you need be able to calculate another kind of frequency. So we've talked about allele frequencies, genotype frequencies, now we're going to talk about haplotype frequencies. So haplotype is a haploid multi-locus genotype. Think of it as like the genotype of a sperm or an egg. Right? So, if you have two genes, an A gene and a big A gene, an A gene and a B gene, A has big A and little a alleles, and B has big B and little b alleles. The possible haplotypes are big A, big B, big A, little b, big B, little a, little b. You say that fast. So they're in combinations in haploid gametes, and that lets you figure out from these haplotype frequencies if selection at one locus, say the A locus, will affect allele frequency at the other locus. And it's actually really easy to do. All you have to do is figure out the expected haplotype frequency, right? And the expected haplotype frequency is the product of the respective allele frequencies. So you would say these loci are in equilibrium if the haplotype frequency is the product of the allele frequencies. So as an example, if big A has a frequency of 0.6 and big B allele at a different locus has frequency of 0.8, what in the population as a whole, how often do you expect big A to occur with big B? 0.48, right? So now if you measure them, if they're anything other than 0.48, then they're in linkage disequilibrium. Right? So they could be found together more likely than you would expect by chance if this is like greater than 0.48, or they could be found together less than 0.48. They're still, either way, it's linkage disequilibrium. And it's really easy to see this because suppose they're found together less than you would expect by chance. Well, that means some of the other alleles at these same loci are found together more than you would expect by chance, right? So it's all one, really. You can quantify disequilibrium. There's a variety of different metrics. One is this D, which is very simple because it's the observed haplotype frequency minus the expected haplotype frequency. And you can then, assess how far from equilibrium are the alleles at these loci. Why is it called equilibrium? Why is linkage called disequilibrium? What would equilibrium be? And why is it an equilibrium? So if this was 0.6 and this one was 0.8, we said the expected is 0.48. And if it's that, then it's in equilibrium, right? If it's anything other than that, then it's in disequilibrium. Why is this equilibrium? What does the word equilibrium mean again? There's no change, and it's the state that a system would return to, right? If these are in disequilibrium, if they're not equal 0.48, they'll tend to return to being 0.48 because of recombination, right? The more recombination you get between them, recombination is what breaks down linkage disequilibrium and returns these haplotype frequencies to their equilibrium state. So that's the relationship between recombination rate and linkage disequilibrium. So again, I should have moved this slide somewhere else. You have to think about this not just as physical linkage, right? It can be due to all these different kinds of things. Any statistical association where these haplotypes are at a frequency different than the product of their allele frequencies, a whole bunch of different possibilities. Right? Here's what I was trying to say. The other slide's in the wrong place. What happens with sex is the recombination of these genotypes that reduces linkage disequilibrium, mixing up which alleles are found with which other alleles. And here's just a graph, the kind of graphs you got from your allele simulation with across 25 generations, the coefficient of linkage disequilibrium as a function of different recombination rates. So what you ought to see, the striking things, right? If recombination is 0, what happens to linkage disequilibrium? Nothing, right? And if it's, the more recombination there is, the faster you restore the equilibrium state where there's not linkage disequilibrium, right? And that's what sex does in a microevolutionary sense. Okay, so suppose they're linked. Suppose they are in. It's kind of confusing terminology. If they're linked, they're in linkage disequilibrium. Does everyone understand that? They're equivalent kind of things. All right, so if they are linked, then you can have selection at one locus affecting allele frequency at a different locus. And this happens most commonly when there's, or one extreme form of this is when there's what's called a selective sweep, where one allele at one locus is strongly favored by selection, so it goes to fixation very quickly. Any alleles at any other loci that happen to be associated with it are going to do what? Not necessarily get fixated, but they're going to increase in frequency for sure, right? So if big A allele suddenly goes to 100% because of really, really strong selection, and statistically, let's say the big S allele at the big S gene is linked to it, that's going to increase in frequency as well. Who knows if it'll go to fixation or not? That's going to depend. So the selective sweep is the direct selection on alleles of one locus, and what's called hitchhiking is the indirect effect on some other allele, some other linked allele. that then increases in frequency as this indirect, it's getting taken along for the ride is the idea of hitchhiking. Okay. Does everyone understand all that stuff?